Sorry. So I'm very happy to have Kenny here again. He gives not only great lectures, but he also has managed to teach monkeys how to play Pac-Man. So in the other social settings, I think maybe you can teach me to play. Maybe not. So let's welcome Kenny.
All right. I'm very happy to be here, but I have to tell you that I only slept for like 2 hours last night because some problem with my flight to Beijing. So I can't guarantee that the things I'm talking about make sense. Just interrupt me. So first of all, I have to claim that I'm actually an experimentalist, not a computational person. But I'm an experimentalist with computational modeling in mind. The reason is that for system neuroscience, anything you do is complicated, right? The brain is a complicated system. If you want to explain how brain works, how brain solves any cognitive problem, you need a model. So the typical routine for us to do research is like this. We will of course first have a goal. What kind of problem we want to solve? What kind of cognitive function we want to understand? Then we train monkeys, or maybe use human subjects to do some behavior paradigm, right? And we will of course measure the monkeys and humans' performance, how they do the task, what's their accuracy, so on and so forth. And then we will try to record brain activity, neural activities, when they are doing the task. Then what we want to understand is how can we relate that neural activity to the behavior so that we can claim certain neurons in certain brain areas is underlying a certain cognitive function. But how do we actually do that if the brain is so complicated and the behavior is also complicated? That's what the models kick in. So we need a model to help us to understand how we can relate the neural activity with the behavior. But what exactly are these models? So the models, so the models I'm talking about here is not like the black box model that many people use. For example, the neural networks. The models here are usually some kind of mathematical formulation of how the behavior task should be solved, based on some kind of optimality assumptions, those kind of things. So the model should be able to fit the monkey or human's behavior well. The model can describe the behavior with set up mathematical functions. And at the same time, the parameters that are in the model should be reflected by the neural activity so that we can say that the brain is actually doing computation, doing the computation similar to what's described by the model to solve the task. So if we can establish this triangle relationship between the behavior, the model, the neural activity, then we can explain then we can claim that the brain is indeed using the computation described by the model to do the behavior task, and we will be able to find out where in the brain what kind of neurons are doing this computation. So this is the typical routine that we use in our research. And today I'm going to talk about two of the best examples, I think, in the field of decision making. How we use the models to to establish the relationship between behavior and your activity. Okay, so I will be talking about two theories. I will start with the simpler one, the signal detection theory, and I will go on to introduce the Drift-Diffusion model. So we have to make all kinds of decisions in our daily life, and the ability to carry out fast and accurate decision is very important for our survival, especially during the evolution. For example, if you are in Africa, you try to survive a dangerous environment. You see there's something hiding behind the tall grass. It could be a harmless deer, or it could be a dangerous predator, right? But the overall the environment is very noisy. You cannot see things clearly, and you only have very limited time to make a decision. So what can you do? How can you make the best and accurate decision to ensure that you are not in danger? That's the kind of problem that our brain has to solve. And we know our brain can solve this problem very well. But the question is, how does our brain solve it? The brain is like the black box. We can record neural activity from the brain, but then we need to find out how this neuron contributes to the decision making. So the signal detection theory is basically a framework trying to help us to estimate how well a system can detect a signal, right? So basically, if a certain system, it could be a brain, it could be a machine, or whatever. The system receives some input, and the job of the system is to tell you whether there's a signal in this input, or the input is just purely noise. So the system just has to do this very simple test. And if we combine the possible inputs and the possible outputs of the system, there could be 4 different scenarios. Of course, when the gratuitous matches the output, the system sends out the correct answer, right? And we can even divide this into 2 situations. When there's no signal in the input at all, it's just purely noise, and the system answers correctly. We would call this type of situation as correct rejection. And on the other hand, if there's indeed a signal, something in the grass, and the system correctly detects a signal, we will call this hit. And also there are and the system could be making mistakes. If there's indeed a signal, but the system fails to detect it, we will call this a miss. If there's no signal, but the system thinks there's a signal, we will call this situation false alarm. So totally, there are 4 possible scenarios, and it's quite easy to see the probability of correct rejection and false alarm should add up to 1, right? These 2 cases happens where the input is purely noise, and the output can only be either noise or signal. So these 2 things should add up to 1, and correspondingly, the probability of miss, that's the probability of hit. These 2 errors should also add up to 1. But the relationship between these 2 are not clear. ideally, you want a system that has a very high probability of hit and a very high probability of correct rejection. But is it possible to build such a system at all? And that's what the signal detection theory try to tell us. And we can frame the problem as this. If the input comes into the system and we have some measurement, we can use a certain criteria to compare against your actual measurement and to decide whether there is a signal or not. And in this simple case, let's assume that the input we get has a normal distribution. So let's say if the input is purely noise, the distribution of the measurement is looking like this. The mean is at this position, and the width of the distribution is like this. And if the input indeed has a signal, the measurement that's produced by the system is like this. And our job, thank you. That's more convenient. Okay, so then our job is to determine when we get an actual measurement without knowing it comes from without knowing whether it comes from this distribution or the other distribution, how do we decide? So as I just mentioned, we can have a criterion to compare against that particular measurement. If the measurement is larger than the criterion, we call it a signal. If the measurement is smaller than the criterion, we call it noise. So by using this criterion, we can also compute how likely we can get a hit outcome, or how, and that's exactly a proportion of the distribution on the right side of the criterion, right? So if there's a signal and the system decides it's and the system's output is also true, so that means the actual input is on the right side of the criterion, so it is this part of the distribution, and the probability that the input sits in this side of the distribution would be the probability you get a hit, and the other part, of course, would be the miss, and you can easily see the probability of miss plus the probability of hit should add up to 1 in this case. But that's the case when the input actually comes from this distribution. If the input comes from the distribution corresponding to purely noise, you will get 2 results. The false alarm is in the case that the input sits on the right side of the criterion. And the correct rejection is the case where the input sits on the left side of the criterion. So overall, we can see how four types of scenario can be plotted on this distribution graph. Okay, so now you can see that because the distribution of these 2 inputs are fixed, we cannot do anything about this. All we can do is just changing the criterion. And if we move the criterion a little towards right, what will happen? The probability of hit will be lower, right? In this case, because the criterion is larger compared to the previous one, so the area and the area corresponding to the hit region would be smaller. And on the other hand, if we look at the proportion of the character rejection, that would be larger, right? It's a part of the red curve, the left side of the criterion. So by moving the criterion higher, we will have a smaller probability of hit, larger probability of character rejection. So that means we cannot simultaneously optimize hit and the character rejection. These two things, although are desirable to us, but within the same system, they actually are against each other. If you want to have a larger probability of hit, you will have a smaller probability of character rejection. So when we want to describe how well a system can distinguish between signal and noise, we have to combine these two things together. What's the probability of hit and what's the probability of correct rejection? And that's what the RC analysis is about. So in RC analysis, we would plot the probability of hit against the probability of false alarm. So false alarm is just one minus the probability of correct rejection. And what we do is we will move the criterion from left to right. And at each level of criterion, we will compute what's the rate of hit and what's the rate of false alarm. And you can see when the criterion is on the left end of the measurement, the system would have a very large rate of hit. And that's because everything would be on the right side of the criterion. But at that time, the probability of false alarm would also be very high. Right? And when we move the criterion toward right, the hit rate will be lower, but the false alarm rate will also be lower. And that's why this curve is looking like this. It gradually decreases until when the criterion is set to very high, then both the hit rate and the false alarm rate will be zero. Okay. And in a different system, let's say for this particular system, the measurements produced by the system regarding to the signal and the noise is very close to each other. And for this system, it's not able to tell apart the two kinds of input very well. And how does the RC curve would look like? It will look like this. So when we move the criterion from left to right, the difference between the hit rate and the false alarm rate is very small. It sits very close to the diagonal, right? So, so imagine if the criterion is here, the right side of the blue curve and the right side of the red curve should be very close because the two distributions almost overlap with each other. So this curve would be very close to the diagonal. So the shape of this RC curve could tell us the performance of the system. And if the system's RC curve is far away from the diagonal, then the system could tell apart two kinds of inputs very well. If the curve is very close to the diagonal, that means the system is not so good. Okay. So, any questions so far? Okay, so if not, then we're going to see how we can use this in the investigation of the brain. So in a series of studies in 90s, starting from 90s, people trying to understand how neurons in the visual system contribute to the visual perception. If we see something, that signal should first come into our visual system, the neurons in our visual cortex would encode the information, and that can be later on be used for judgment. So how these neurons encoding would affect our perception? the particular behavior paradigm we use to do this study is called the random dots motion direction discrimination test. So in this test, the monkey is looking at a bunch of dots moving together. So a bunch of random dots moving, and only a proportion of the dots are moving coherently in one direction toward either left or right. And those dots are illustrated by the black dots here. The other dots are just moving randomly. So the black and white are just shown for the purpose of illustration. What the monkey actually sees are just uniform white dots. The monkey did not know what the proportion of the dots is moving coherently. And their job is just to tell you whether overall these dots are moving toward left or right. Okay. And I will show you an example. So if you stare at the screen, can you tell me the directions of dots? Right, yeah. So that's actually the case where all dots are moving to our left, and that's quite easy for you to tell. If adding some noise, you can see now the test becomes a bit harder. Okay, let's try this. You see? Okay, I'll do it again. Right, not so easy, right? But so here, about half of the dots are moving toward your right, and for people who are not used to this kind of stimulus, it appears to be hard. But it's actually quite easy for monkeys to do this test, this level of noise. And we can see more examples of even harder. It's right?
All left.
Okay, that's right. So this is approximately the level the monkey's performance starts to drop below 80%. Okay, it's also hard for monkey, but let's see an example of a very hard situation. Right.
All left.
What would be your what would be your confidence in your answer?
Mine is like 95.
Really? Wow. In this case, actually, that's a very it's a trick question. But the monkeys would also do that. So the monkeys would be staring at this stimulus and trying to guess based on just noise. So if we plot the monkey's performance, you can basically see is what you would expected. The monkey's performance would drop from almost perfect to chance level when the correlation or the coherence of the moving dots drops from 100% to very low, right? That's not surprising. Then what people try to understand is this performance, can we explain this performance by looking at how neurons in the visual cortex encode the motion information. So to address this question, people are looking at one particular area within the visual system called MP. And this area sits in what we call the dorsal stream of visual processing. So for people who are not so familiar with the visual system in the brain, first of all, this is a monkey brain. Here is the frontal area, so the anterior part, and here is the posterior part. And the visual information coming from the eyes first enter our cortex in U1. So it's a bit counterintuitive. the eyes are different, but the information first goes to the back of the cortex. And starting from V1, actually, the information would go in two directions. And in what we call the dorsal stream of visual processing, it deals with information of movements and spatial information. And MT is a very important area in processing this kind of information. So naturally, for this random dots motion direction test, people would think MT would be an important area to look at. Then they trained the monkey to do the test. The monkey can do the test, and then they recorded the neural activity from the MT area. And here's what they find. So this figure is a bit complicated, and I will try to take you through. So here you see five figures. Let's look at the top figure first. So the top figure is a case when the monkey is looking at a dot pattern with a coherence of 12.8. So at this coherence level, the monkey is still performing quite well. And the bars you see are the distribution of the neurons responses. So for this particular neuron, you can see if the dots are moving toward left, but yeah. So if the dots are moving toward the left, the distribution of the response produced by this neuron is like this. So roughly, the mean is wrong. 30 or 40 spikes per trot. And that's a measurement produced by the system. If we relate it to the signal detection theory framework. If the dots are moving toward the right, that's the distribution plotted here with the white bars. So the mean is much higher. And overall, these two distributions are separated quite well, right? So let's imagine in a particular trial, this neuron has a response of 80 spikes per trial. And you can be pretty sure that response comes from this distribution, right? It has a very high probability of this distribution. So if you just use the response of this particular neuron alone, you should be able to do the task quite well to tell apart whether the dots are moving toward left or toward right. Then what would happen if we make the test easier? If we make the test more difficult? If we lower the coherence to 6.4, you can see the two distributions starts to overlap more. Now the mean of the response for the Y distribution is closer to the mean of the response of the black distribution. And the same trend goes on if we further lower the difficulty until when the coherence level is very close to zero, the two distributions almost overlap with each other completely. And at this point, you basically cannot use the neuron's response to do the test at all. If you get a response, let's say at 50 spikes per trial, you wouldn't be able to say confidently whether it comes from the or the black distribution, right? So based on these measurements, we can plot the RC curve that I measured, the RC curve that I mentioned earlier for each coherence level, and they will look like that. For the coherence level of 12.8, the curve looking like this. It almost sits on the side of the square. That means the probability of hit and the probability of false alarm can be well separated. It's far away from the diagonal, and the system performs very well in this situation. When we lower the coherence, you can see the curve moves more and more. We can see the curve moves closer and closer to the diagonal until when the covariance is at 0.8%, the curve almost sits on the diagonal. And in this situation, the system is almost useless. And furthermore, based on this RC curve, we can calculate what we call the neurometric curve. And that's what we will be, that's how accurate we can do the task just based on this neuron. And this curve looks very similar to the behavior curve that I just showed you. It's also, neuron can also do very well when the coherence level is high and do poorly when the coherence is low. So by these results, we can say that maybe this neuron can reflect the performance of the monkey. Actually, if we compare the performance of this neuron and the performance of the monkey, you can see the shape does look very similar. Yes, please.
How do you calculate the specific way the RNC?
Oh, right. I didn't mention how exactly we get this correct. Yeah. So here with this RC curve, we can quantify how far away these curves are from the diagonal just by calculating the area under the RC curve. Okay. So for example, for this curve, the area is very far away. The curve is very far away from the diagonal, and the area is almost 1, right? And the system can perform very good. But for this curve, it sits on the diagonal, so the area is basically 0.5, right? And we can use this number as a measurement of how good the system is, and that's what's being plotted on the y-axis. So the RC curve is a way to measure the system performance even without setting a criterion.
Okay, back to last slide, please.
Yes.
Yeah.
Okay.
So in this slide, if you have two distributions, I guess the black distribution is going to give that sigma.
Right.
The other one is right. And there's some overlap, right? For those trials,
Right.
what happens to a monkey? Is the monkey complete or?
Yeah. First of all, the monkey's performance is not 100% correlated with the neuron's response. This is just one neuron, right?
Yeah.
There are like millions of neurons in the brain, and for any particular trial, maybe the neuron's response is this, and the monkey says right. Yeah. So, but statistically speaking, that when the neuron response is on this side, the monkey is very likely to say that.
And question is on the next slide. Right. On the right figure, and how do you actually get the why is it more like y function?
Right. I actually don't remember what curve they used in the original paper. But yeah, I need to go back and look at the paper. But I believe this figure, this curve is arbitrarily chosen. So it's some kind of a function that has this S shape. Yeah.
In this ROC theory, so the assumption is that somewhere in the brain there is a scalar, one scalar, and I choose if it's larger than something, I choose this, and if it's smaller. But is this an assumption? Because I can imagine that
I don't know, I have multiple numbers and I choose according to them, or in this regime I choose this, but in this regime I choose something else, and then it switches. So I'm trying to understand if this
Right.
this is an assumption or it's
Yeah, so first of all, this kind of analysis is only used in scenario when there are only two possible answers. Okay, so if there are more than two possible answers, you cannot use this RC. And secondly, there is a theory behind that. In that sense, this would give you the most powerful test. Yeah, to tell this alternatives apart. Right. So, right. Yes, let me let's see if I misplaced the figure somewhere. Oh yes, that's figure. Yes. So I mentioned that now we can have the monkey to do the test, measure the monkey's performance, how accurate the monkey is. And simultaneously, we recorded from the neuron data, and we can use the neuron activity to calculate how good the neuron can be if we just use the neuron activity to do the test. And that's what we call that's what we call the neurometric performance. And we can compare these two things session by session, right? So for each data point here, that's one neuron. So for this particular neuron, we can calculate its performance. So here what we plotted here is the threshold. So the threshold is basically the coherence level at which the neuron can do reasonably well, and we can use the same threshold to compare how well the neuron do and how well the monkey is doing in that particular session where we are recording from this neuron. So we can do this. So we can repeat these experiments many times and get this plot. And we can see very, very interestingly that it looks like all the data points sits quite close to the diagonal, with the mean very close to the diagonal. That means, on average, the neuron threshold would match the monkey's behavior threshold very well. So, in other words, we can explain the performance of the monkey by how neurons from the empty area encode the motion information. Okay.
So single neuron, so where's the second neuron?
Sorry.
I don't know, so it's where's the second neuron?
No. Each click point is a different neuron sampled from a different session, different day. And
The different colors like
so here, different colors are just a significant test between the two thresholds. So the dark is there are no significant difference between the behavior and the neuron threshold. Yeah. And that's actually a very surprising result, right? So here we're comparing the performance of single neuron against the performance of the monkey. But the monkey has like millions of neurons in there. Why would the performance of the single neuron would match the performance of the monkey? Does the monkey really just use one neuron to do that test? Actually, we don't know the answer until now. And there are many explanations. It could be a coincidence finding here, because we know that a population of neurons should be able to encode information better than single neuron, right? But at the same time, there are many more processing levels after NT, which could lose information, and there might be other cognitive factors that contribute to the monkey's performance that's not accounted by the encoding ability of the monkey, encoding ability of the neuron. So, so what they found here, I would say, does not mean that monkey is using just one neuron to do the task. That would be absurd, right? But it does tell you that we can relate the neurons, we can relate the monkey's performance to the neurons encoding capability, and their relationship, and we establish the relationship with the help of the signal detection theory. Okay. Any questions about this part? If not, let's move on. So, so it seems like the signal detection theory would be a very useful framework to explain how monkeys or how we can explain the monkey's performance with neurons activity. But you might notice there's something missing in this framework, which is time. What do I mean by time? Let's go back to the example that I asked you guys to do. So in this example, the task is quite easy, right? It's 100% coherence, all dots are moving in one direction, and you can have the answer in a very short period of time. And that's the time I'm talking about. If you are looking at a harder trial, in which case this is 12.8, you would have to spend more time to decide whether it's left or right if you can even get an answer, right? So for us to make a correct direction, to make a correct decision, it actually takes us a different amount of time depending on how hard the task is. And that's the part that's missing from the signal detection theory, because it's not telling you anything about how much time you would have to spend on your decision. And that's what the Drift-Diffusion model can see. So, wait, sorry. So to save time, first of all, we would need a behavior paradigm that also reflects time. So in the previous version of the task, we just let the monkey to watch the random dots for 2 seconds, and then ask the monkey to judge the direction of the motion. But now we change the behavior paradigm a bit, so that the monkey can decide how long they want to view the motion. Right. So in the beginning, the monkey would stare in the center of the screen. Then at certain points, the motion starts, and the monkey can take all the time he wants to decide whether the dots are moving toward left or right. And finally, the monkey uses the eye movement to report their decision. If it's right, the monkey will stare at the right target on the screen. And the time between the onset of the motion and the time the monkey moves its eyes toward the target is what we call the reaction time. That's how long the monkey needs to tell to do the task. And first of all, let's look at the behaviors. And as you would expect, the accuracy of the monkey in this version of the task has a very similar pattern to the previous one, which is the monkey does very well when the coherence is high. The monkey is practically guessing when the coherence level is very low. And the new thing is here. As you would expect, the monkey's reaction time is much longer when the difficulty is high, when the coherence level is low. And the monkey is much faster when the difficulty is low and the coherence is high. Right? That's very natural. That's what you would expect. So how do we explain this? So, so this is what the Drift-Diffusion model is for. So in this model, we will have so the decision process in this model is described as a drifting process of a particle between two thresholds. So the particle would start from a neutral point to the 0 point, and as time goes on, and the information comes in, the particle would start drift toward one of the decision threshold. And if it reaches, let's say, the top threshold, it would say it's decision A, or in that version of the test, it would say left. And if the particle eventually ends up here, the decision would be in favor of the other. But how fast the particle drifts is decided by the evidence. So the evidence is modeled as a noisy, as a random variable with a normal distribution. So the mean of the so mean of this variable is determined by the signal in the inputs, and we call this momentary evidence because this input would vary from time to time, right? And of course this signal is noise. That's why. And the noise is described by an invariance term here in the normal distribution. So you can see that overall, because the mean is not 0, so the drifter should be biased toward the direction that's determined by the sign of the mean here. If the mean is a positive value, the drifter should be biased toward the top threshold. But because it's noisy, so even if the mean is positive, sometimes the particle will still end up in the opposite direction, and that's the case when the system makes a wrong decision.
I guess I have two questions. The first one is momentary evidence changing? Well, is this one like exciting based on time t?
Yes.
And then secondly is this feels like it's possible to extend it like it's experimentally or
Yes. But it's actually mathematically quite difficult to extend it to more than two alternatives, because you can imagine if there are 3, then the drifter will no longer be in a plane, right? It would be in a high dimensional space. Then in a high dimensional space, the form would be described line. There are actually many different kinds of models to set up the multi-alternative cases. But for the interest of this course, we will just focus on the simplest case when there are just 2 alternatives, and the drift only within this plane. It doesn't have to, yeah, but here I'm just showing the simplest case when it's symmetric. So you can imagine if it's not symmetric, then the particle would be more likely to hit the threshold that's closer to the starting point. Then the system would have a bias, right? So asymmetric thresholds can be useful when the N mode does have a bias in its sense. So, yeah. So, so with this model, not only we can model how accurate the system is. That's a probability. That's a probability of how often the particle will hit the correct bound. And in addition, we can model the reaction time. That's how much time the particle takes to reach the bound.
Sorry, I might be jumping the gun here. But how much of, I guess, like you're saying about that, so like how much is the change? That just like, I guess, like, that to the question of this particular problem, because it would sound tough.
I don't completely.
I just jumped off the previous point about like there could be biased in our things that I think it's like biologically how it's balanced.
I will talk about this later. But right now we are not talking about neurons yet. We are just talking about in general how the model is. Yeah. So so with this model, not only can we explain how accurate the system is, we can also explain how fast the system is when the system makes a decision. And you can imagine that reaction time would depend on the quality of the evidence very much, right? So if the system has a smaller, if the input has a smaller mean, then the evidence is not very strong. It's actually the opposite. So if the evidence is strong, it has a larger mean. That means the drive toward one of the bound will be stronger, and the particle will take less time to reach that. Right. So that's a case when the machine makes fast response to an easier task, and in the opposite direction, if the evidence weak, it's very noisy, with a small mean or a large variance, then the particle would take much more time to reach the bound, and that's the case when the reaction time is long. So even without any mass, you can see the model would make sense to account for what we intuitively see the relationship between the reaction time and the task giving. But actually, we can do math to compute what exactly the reaction time, the accuracy of the system is, if we know the distribution of the momentary evidence and also the height of the bound. But I'm not going to do that step by step. I'm just going to show you the equation. So the probability of hitting one of the bound, the probability of hitting the correct bound is a function looking like this. It's both the function of the bound height, A is the bound height, and the mean and the variance of the distribution of the momentary evidence. And you can see if the bound height is higher, the probability of hitting the correct bound would also be higher, right? So if it's larger because of the minus sign, the exposing will be smaller and the probability will be higher, right? And on the other hand, this is the reaction time. Again, it's a function of both the bound height and the parameter for the distribution of the momentary evidence. And again, if the bound height is higher, it will take longer for the system to reach the bound. So the reaction time will be also longer. And so So you can see there's a trade-off between the accuracy and the reaction time. So to have a higher accuracy, the reaction time would also have to be longer. So if you spend more time to make your decision, you can be more accurate, right? That's quite reasonable.
So what is ET?
Oh, ET is the mean of the reaction.
Also see this kind of P probability looks right. So the max in space of the and we regard the term in the exposure operates as some kind of level function.
That's interesting thought. Well, well, I need to think about it. If you look at this way, how do they explain everything? But this is but you are correct. This is essentially a function, mathematically. I'm not sure how to relate this to the value that you're talking about. So now we have these equations.
Other questions?
Yeah. So the question is, this Drift-Diffusion model is kind of like one-dimensional random walk depending on the probability distribution. What bother to do this model? Why not just model the reaction time using a distribution?
You think of distribution of what?
Like reaction time.
Yes.
And maybe there's a probability distribution for correct?
Yeah. Yeah. But yes. Yes. That's actually a very good question. Why we bother to model everything like this? And that's because we want to relate this to the neural activity in the brain. That's what we actually see in the brain. I will show you later. So all the model we are doing is because we want to relate the behavior to the neural activity. The model serves as an explanation why we see the neural activity as this. Okay. So now these equations, now we can use equations to fit the monkey's behavior just to see how well the model works, right? This is just the old theory that what could be how you could be solving this test, but we don't know if the monkeys are actually using this to solve the test. And you can see actually the model works very well. So here the data points are the actual data points we get from the experiments. The monkey's accuracy versus the motion coherence and the monkey's reaction versus the curve are the functions from the previous equation. So, well, with the curves as it fits using the previous equation to this data points, and you can see they match very well, right? Almost perfect. And I have to mention that because these two functions share the same variable, right? So we can first use only these data points to fit one equation dot all the variables, and then we use those parameters to produce the curve for the other for the reaction time data set without really fitting the reaction time and seeing how the model works. And this is what we can get. So with just the accuracy data points, we can produce a reaction time curve very well. That means really the model can simultaneously explain both the monkey's choice, accuracy, and the monkey's reaction time.
So how exactly did you fit the model? In practice.
Oh, This is just the very standard functioning thing. You have a lot of data points, you calculate the likelihood. There are many different ways to do this kind of thing. For example, by using gradient descent or this.
You know, expectations.
I'm not exactly sure, but I think it's just a very standard maximum likelihood kind of thing. Yeah. Okay, so now we see the model can explain the behavior very well, and I also just said that the model is created also to explain how the neurons, how the brain solve the task. Now let's see how the brain solves the task. So I previously mentioned that the neurons in MT encode motion information that can be very useful to solve this task. And now we are actually moving on to a different area called LIP. So LIP stands for lateral intraparietal cortex. That's a brain area within the parietal area. Also sits in the dorsal processing stream of the visual information. The reason we choose LIP is because it has some interesting property that's not shared by MT neurons. And what are these? So I mentioned some details. Maybe you can pay attention to. Is that for this particular task, we estimate to use the eye movements to do the task, right? So at the beginning, the monkey is looking at the center fixation point on the computer screen. Then some random dots pattern would appear in the center. Then whenever the monkey is ready, it would use its eyes to tell us its position by moving the eye either to left or right. And the interesting about LIP neuron is that it has something what we call the response field. So here the response field is indicated by the gray shading region here. So for a particular neuron, it has a response field. It means whenever the monkey is going to move its eyes toward its response field, the neuron would become active. Okay. So when the neuron, if the monkey is moving its eyes to anywhere else, let's say the left target, the neuron will not be active at all. That's what response field means. So the reason we want to look at the LIP neuron is that because it tells us what the monkey's final position is. In addition, there are some studies showing you that they will do that even before monkey starts to move its eyes. Then we wonder, would this neuron tell us the decision that eventually produces eye movement during this time? And here are the results. This figure might be one of the most famous figures in the field of decision making. So I would like you to really pay attention to this figure. They say it's from the review paper published in the 2007. It's not the original research paper, but I think the review paper version is better for the illustration purpose. So here is the response of LIP neurons. The y-axis is the firing rate, the x-axis is the time. 0 is the onset of the motion pattern. So when the monkey starts to see those dots, you see the LIP neuron have this kind of activity. Each trace represents a case of a particular motion coherence. And the solid lines, and the solid traces, and the dotted traces are the two situations when the monkey either chooses the target within the response field of this neuron. That means the monkey is going to choose a target that the neuron likes and would activate this neuron. That's the solid trace. the dotted trace is when the monkey is going to choose the other target outside the response field, and the neuron should be silent in this case. Okay. And let's just look at one of the curves first. So for this orange curve, it's a case when the coherence is high, 51.2. The task is really easy, right? You can see the response of LRP neuron after the motion onset will quickly rise in this case. It will go to a very high level in a short period of time. If the motion strength is lower to 12.8, you can see the overall pattern is actually quite similar. The neuron will still have an increasing fire rate over time, but the increase seems to be much slower comparing to the case when the coherence is higher and the evidence stronger. If we even if we look at the evidence level even lower, let's say if we take it to the stream 0 coherence, that's the case when the input is completely noisy, but also the monkey still chooses a target that the neuron likes. You can see still an increasing fire rate of this neuron, and again now the increase is even slower compared to 12.8 cases. But for the opposite direction, if the monkey chooses the target outside of the response field and the coherence level is strong, you can see a very quick drop of the response of these neurons. And the drop is slower when the evidence is poorer for the target outside the response field. And more importantly, by the time of the decision, this when the monkey starts to move its eye toward the target, you can see all these traces will merge together, right? And what does that mean? That seems to be very similar to what the Drift-Diffusion model said. When you make a decision, your accumulated evidence, I mean the particle, the drifting particle should reach a threshold, right? And that's what we see here. At the time of decision, the neuron's response seems to be reaching to a threshold. And the opposite case is that when the monkey decides to go for the target outside the response field, you can see the neuron goes downwards.
Just checking to make sure I understand the difference between the dotted line and the full line correctly. So the full line is if the motion is to the right, if the dotted line is to the left. Is that right?
Or not? Is it not exactly? So the solid line is when the dots, when the monkey chooses the target that the neurons like. The target sits inside the response field, and for the high coherence case, that's most likely also the direction of the dots movement, right? Because in that situation, the monkey is almost always correct. But for the 0% coherence, that's like half and half. The dots are moving totally random. Sometimes the monkey gets the right, sometimes the monkey gets the left, right? But the solid curve here means the monkey chooses the target that the neuron likes. And you can see the solid curve and the dotted curve in these cases have exactly the same input statistically, right? But the neuron's response are very different in this case. In one case, it goes up if the monkey chooses the target the neurons like. In the other cases, the response goes left. So here on the left side, the response is aligned to the motion onset. You can see the separation of the neuron's responses if we divide the trials by different level of motion strength. But when we align to the eye move, when we align the responses to the eye move, even in the case of 0% covariance, eventually we'll go to the high point before the go to the high point above the time when the monkey is to make the decision.
There's something special about this high point, central here.
Yeah, something special about this time point. This is a time point when the monkey is making the decision.
High point.
Yeah, here. Yes, that's the point when the monkey is going to make its decision. That's
This question again, no, he's asking like, is that value of 67?
oh, so you're asking whether this particular number means anything.
Right, because they're always the same value.
Yes. Oh, well, we don't know. That's just what we get from the data. There seems to be, well, first of all, this is the average from any neurons we recorded from the brain. We don't know if for each neuron, it has a particular number that corresponding to the neuron's threshold. And we also don't know if the neurons has to collectively hit the threshold for the machines to make a decision, or it's just some of the neurons has to do.
Because experiments.
Yes, yes. This is actually one of the hotly debated area in the field. So there are conflicting results. Some experiments showed that if you stimulate these neurons with microstimulation, you can change the response of the neuron consistent with this model. But sorry, yes, and but there are also other studies showing that you cannot. So right now it's not conclusive. But what's considering found is that the activity kind of will match the behavior of everyone.
Think about the neural stimulation also being observed in other brain regions such as also the PSC. So how should we think about the role of LIP in this discussion?
Right, that's a very interesting question. Yeah. So here I'm just showing you that the activity pattern of LIP neurons is not very well. So that alone does not say that LIP is the only area that's doing this decision. As you mentioned, actually the similar pattern has been found in many different places in the brain. And right now we don't have a definite answer. It could be the case that all these brain areas are making decisions simultaneously. There's redundancy in the system so that it can make sure the system would work if when not the brain is damaged. Or maybe different system would serve different purposes because we only use this particular behavior paradigm to look at the to this investigation. It could be the case that different area would be more suitable for different type of tests. That's also possible. And another possibility is, of course, this system sits on that decision-making train. One of the area is doing this calculation first, then it passes the signal to a different area. But right now, the evidence I would say it's against this theory. But it's still a hotly investigated topic in the field.
Were the movies in this experiment? So the activity is like perfectly aligned to the item, but the experiment was still that the monkeys were not told when to respond, they could accumulate as long as they went.
Right.
See.
would record from some motor neuron controlling the eye, would you expect to get kind of similar results?
Yes, yes, that's a very good question. So if a neuron is purely motor, you would expect that the neuron would only have response near the time of eye movement. It shouldn't bother everything.
But if there is some preparatory activity, so you also
Yes, yes, that's actually what can be found. So in premotor areas, people what state they are mostly motor signals. You can find very similar things into this, and the question comes back to what's our relationship between all these areas. This is the first study that found the neurons in the brain that matches the DBM very well. So ARK is the first area where people find such activity. It's not the only area. And now there are many new studies showing the same activity that is observed in many areas. So the answer is still out there. How to interpret this result.
So I have a question where diffusion rate. This basically you are the motion offset, the activity is light, right away after. Like the diffusion rates are different.
Right.
I wonder what's the mechanism of that.
Right. So the zero is the time of motion offset, and I think it's very reasonable to assume that the motion information takes some time to arrive to this brain area, and this period could be the time that that takes them. Could be the time it takes for the motion information to arrive there. And until this data point, maybe you can see a different. You can see a difference in terms of their activity without different levels.
It's also
Yes.
that's the upstream. And T, you know, with different components.
Yes, that's actually what I have talked about in this part. Yeah. So people also simultaneously recorded on the MT area, and I can see the pattern in MT area is very different. Here, the MT response seems to be a sustained response up to motion offset. So if the motion strength is high, the neuron is a sustained response, higher response. If the motion strength is low, the response is lower. So that's a very different pattern compared to this. And that's consistent with the idea that MT provides a momentary evidence here, right? Because in this particular task, the momentary evidence does not change during the streaming, right? So the evidence strength stays the same throughout. So the activity of the MT seems to encode the quality of the evidence. Here we see a random pattern, either up or down, depending on the motion direction, and that's more consistent with the particle drift into the end.
During the movement of the eyes, those neurons won't be active. During the movement of the eyes,
Okay.
those neurons won't be active.
Oh, well, so in this version of test, during the movement of the eyes, the random dots is shut off. So the monkey will not be looking at the dots anymore. And this neuron typically have this response pattern. If the eye starts to move, the activity will jump.
question more like, is there are like neurons that are recruited only for decision and other neurons that are recruited only for execution?
That's true in the other brain area. So for example, if you go to downstream model structures like supercalyptus, you can find neurons only responsive for the eye. You may argue this is only important for the execution of them. But for the LFT neurons, most of them have some response during the time when it's trying to make decisions. Yes. People have the experiment seen in MT and find that if you stimulate change its direction according to what kind of your to be activated. Yes. So the role of MT is more confirmed compared to LRT in this sense. Yeah.
When we stop the movie, yeah, it's superficial. What happens?
Right. So if you stop the movie before the mechanics decision, that means there will be no new evidence coming in, right? Then you can imagine it's like you are receiving zero mean inputs into the system. Then the rest of the decision is just the noise. But it will still take some time to commit to a decision, but the decision would be, but the accuracy will be much poorer.
Okay, those neurons are excitatory.
And when we're doing this kind of recordings, we cannot tell whether the neurons are excitatory or not. But we believe most of these neurons are excitatory. Okay, so the summary of the second part. How much time do I have? 5 minutes. Okay, that's too bad because I still have a lot of time. So that's a quick summary. So the Drift-Diffusion model provides a framework for us to understand both the choice and the reaction time in decision making. So see how am I going to spend those 5 minutes? Well, I think we can just stop here. I will be part of the rest of the talk to my next talk. So I will just quickly point out the DDM model and in general the evidence accumulation process during decision making is actually seems to be a general mechanism how brain makes decisions. It's not only for the kind of perceptual decision making that I just showed you. It's also used in value-based decision making or time judgment. Actually, the first papers regarding to the first papers using the DDM to model decision making is actually to investigate memory retrieval. So when we have retrieved memory, that can be also a process of integrating noisy memory over time, and also probabilistic reasoning and the other things. Okay, so So we have 5 more minutes for question.
These references here are mostly for binary tasks. I'm curious whether or not, just kind of trying to do this randomly, but probably someone has tried to do like a chain of these tasks, or at least like more than one decision. So you want to get the stimulus and has to make 2 choices, for instance, maybe a event or something like that. Or ideally something like that, 2 choices of the same nature. And I'm curious to what extent this framework is consistent with that type of experiment, or what the result would be. Sort of, if it's hard to do that type of experiment.
Yeah, well, first of all, in terms of doing experiment, it's actually not very hard. You can imagine, let's say in the dots experiment, you can just set up 4 targets on the screen and having the dots moving in 4 possible directions and estimating to do the task. That has been done, and the people have also found a similar ranking up pattern. There are many interesting subtle features in those multi-alternative kind of experiment that people see.
I guess the important point here, though, would be that there you can imagine that if they're all independent, it's kind of the same as doing separate trials, right? Mm-hmm. But importantly here, I'd be interested in what happens when there's a relationship between the 2 decisions.
Yes.
Maybe the first one has to be opposite of the prior one or something.
Exactly.
It's the same result.
No. That's quite difficult for multiple reasons. So, so first of all, theoretically, it's very hard to set up a DDM type model in the high dimensional space, as I just explained. So there are many different ways to set up the DDM in high dimensional space, and none of them has been completely successfully to explain the behavior of the monkeys or humans. And secondly, for types of experiment like random dots, it's actually very difficult to control how the evidence what correlates between different choices, right? So in the case of the true alternative, the left versus right, that's 2 very different directions. But when you go to like 4, then you can imagine some neurons might simultaneously encode motion directions upwards and downwards. Right? So this neuron would contribute to both choices. Then naturally, the evidence received received by the downstream neuron would have some correlations, because in this case, the 2 directions would be contributed by the same neurons. Will be contributed by some of the same neurons, and there are a lot of complications in terms of things that cannot be easily solved by this type of behavior task. But people have been trying with different kinds of setup. So the short answer is it's difficult. People are doing that. We don't have a very good answer here.
Maybe just really briefly, if you remove the high dimensionality of the output space, just maintain a binary choice, right, but have the 2 sequential things be related, like the animal has to reverse the contingency or something like that, or some kind of, it has to maintain an average in prior trial, essentially.
Right. Then
what happens? Is it untenable experimentally, analytically, or is it people have tried and haven't tried?
People have tried, but as you can see, with all the complex problems that I just mentioned, there haven't been any clean answers so far.
I know there are some more evidence accumulation models like the key computing model. looks just like that system. So I think can it be linked to like differential equations and to model the neural data and like the neural itself outputs the decision variables.
Yeah, there are many different ways to model the behavioral system. So I guess you are probably mentioning models like the attractor networks, right? And for this type of models, actually it does not really tells you the mathematical principle to solve the problem here. So the beauty of the Drift-Diffusion model is that it's backed up by a statistical procedure that's proven to be optimal to solve these two alternative choices problems. I was planning to introduce this in this lecture, but I will talk about this in the next lecture. Yeah, so I think that's what's nice about the DDM model. It's completely interpretable. Every component of the model have some explanations. And when you find the neural correlates with the components of this model, you know well, or you can explain well what these neurons are doing.
So in the Drift-Diffusion model, there's a boundary. And sometimes like speed, speed accuracy, that's how tight the boundary is.
Right.
So I wonder if there's any neural correlates of this boundary and how that.
That's a very good question. How to get this one? Yeah. So the thing about the boundary is actually quite difficult in terms of finding the neural correlates. The reason is that the boundary actually the reason is that it's impossible to dissociate the boundary with the drift rate, right? Because the two things can be scaled. If you increase the boundary twice, it's the same as you lower the drift rate by half, right? So when you're trying to find the neural correlates, if you see the neurons have a lower response, you do not know exactly whether it's a boundary change or a drift rate change. Experimentally, what's typically being found is that the neural boundary does not change by much. So when the animals are slower, or when the evidence is weaker, or in general the speed accuracy is different in the animal's behavior, we do not find the neurons boundary changes very much. So that's the general finding, but we do not know if that means the brain is setting up a different boundary, or the brain is or the neurons are using a different drift rate to do the decision.
So speed accuracy is changing noise level? Or...
That's an open question. We don't know how it's implemented in the brain. Yeah.
We can not only make central decision making, we can also create or adjust our behavior according to the customer. So I'm wondering how to deal with the flexibility by using.
Yes, that's a very good question. I'm also going to talk about this issue in the next lecture. But the DDM model. So the short answer is that the DDM model is actually quite flexible, right? The evidence comes in. Does not have to be perceptual evidence. It could be also the evidence in the other domain, like value or memory, for example. So for particular behavior tasks, you can set up the model so that the evidence So the drift rate reflects the evidence used in this particular behavior test to do to use this model to get the behavior. And sometimes the evidence could be completely abstract, like in the case of memory retrieval, we don't exactly know what noisy memory evidence is, right? But we can still use the model to do the fitting to figure out, like when people are slower in memorizing something, when the people are slower in recalling some past memory, then the quality of that memory is poorer, although we don't know what exactly that means, what's the neural signal regarding to that memory is.
It was a great introduction. So, the model these models will very nicely the link reaction patterns and the probabilities. But what about the distributions exactly? You have a lot of data. So, in our experience, you need collapsing bounds to model well the shape of the distributions. And you know also that collapsing bounds are going to be the optimal strategy when you don't know the coherent, right? So the DDM model, like the flat boundaries, is the optimal one if you knew the coherent. Experiment that doesn't happen, right?
Right. Of course. There are many variants of the DDM model you can change. Yes. So what Ruben is mentioning that maybe the threshold does not have to be constant across time. You can imagine if the threshold is constant, that means you would have to take a very long time to reach a division if the evidence is very, very weak, right? But that normally does not happen when we are facing a very difficult question. We will still eventually reach a division within a reasonable amount of time. So to model this, people have created a model in which the decision threshold would become smaller across time. That's what we mean by collapsing bound. And with this kind of model, we can with this kind of change, we can model the case that people are still taking a reasonable amount of time, even when the evidence is very, very weak. Yeah. So. Okay.
If everyone's hungry, especially me, so let's go to Tianming again. Okay. 谢谢大家，辛苦我了。